Yes, good evening, students. Today we start with the genetics and evolution, that chapter. That is a brief account of this chapter, mainly the theory portions. First chapter, that is principles of inheritance and variation. Inheritance of variation. We start with this chapter and the brief account of the theory of this chapter. It is very important. And that chapter that is a potential source of questions for Neet examination. There will be 5 to 7 questions or 5 to 7 questions or 5 to 6 questions it will be asked from this chapter, that is called principles of inheritance and variation. And this unit, genetics and evolution, consists of 3 chapters: principles of inheritance and variation, molecular basis, and evolution. And we have to go through this principles of inheritance, the chapter. What this chapter deals with. And before that, two scientists will introduce, that will be James Watson and Francis Crick. That is Watson and Crick, and their important contributions and their PhD thesis and their contributions, they are all given, and they get Nobel Prize, all over the Nobel Prize in the 1962. All these things you have to go through this text part. That is a very important part, and from this also the questions will be sometimes it will be asked. That is not sure, but it will be also important. James Watson and Francis Crick. Okay. Then next, the principles of inheritance and variation. This chapter deals with that is mainly the branch of science that deals with the heredity or inheritance and variation. That is called genetics. The branch of science that deals with the inheritance and variation. That is called genetics. And what is meant by inheritance? What is meant by inheritance? Inheritance means the set of characters that will pass from one generation to next. One generation, from parents to offspring. That is called inheritance. And it is the basis of heredity. It is the basis of heredity. And what is meant by variation? That is in the case of the degree of variation. The degree of the offsprings to differ from parents. That is called variation. But that is the 2 important features that we study in this chapter. That is inheritance and variation. That is very important. What is meant by inheritance? The set of characters that will transmitted from one generation to next. It is called inheritance, and the degree of differ from the progenies to parents is called variation. But that is very important 2 features that is connected with the heredity and variation, or inheritance and variation. And who was the father of genetics? Father of genetics was Gregor Johann Mendel. Okay. Then human know early as 8000 to 1000 BC, one of the causes of this variation was hidden in sexual reproduction. That is, during this our prehistoric man, at that period 8000 to 1000 BC, the causes of variation was hidden in sexual reproduction. Okay. And these variations they are exploited in the wild populations of plants and animals. Wild populations, or wild animals and wild plants. They will do selectively breed and select for organisms that possess the desirable characters. That is the process called artificial selection and domestication. That process is called our prehistoric men at that time also they will know this artificial selection and domestication. That is because the variations are hidden in sexual reproduction. Then they will do this in some ancestral wild cows. And in India, we developed an important cow that is called Sahiwal cows in Punjab. It is domesticated. Artificial selection and domestication, they will develop a well-known Indian breed. It is called Sahiwal cows in Punjab. Okay. Then next, another important feature that is in the case of our prehistoric men. They will recognize that our ancestors knew about that inheritance of characters and variation. But they had very little idea about scientific basis of these phenomena. They have no little idea about the scientific basis. They will know something, it will be takes place during this sexual reproduction. There will be hidden some features or variations that will be hidden in the organism that will be in the wild plants or animals, and they will select and breed and produce new breeds, and they are called domestication with human care. That is called with human care. It is called domestication. That is improvement of these plants and animals from wild plants and animals by domestication, by human management. Human management, it is called domestication. Okay. Then next at the time being, there will be laws of inheritance will be published by Mendel. Mendel's laws of inheritance. That is very important and it is highly accepted, and it was takes place during the mid-19th century. Mid-19th century, it is the headway was made understanding of inheritance. Grigor Mendel conducted hybridization experiments on garden pea for 7 years. 7 years. That duration is very important. 1856 to 1863. 7 years. That will be asked in what that duration is asked in one NEET examination. Okay. 856 to 863. 856 to 863. Okay. And proposed the laws of inheritance in living organism. That is the case of he proposed the laws of inheritance in living organism. Okay. During Mendel's investigation into inheritance patterns, it was for the first time he will do the statistical analysis and mathematical logic. Statistical analysis and mathematical logic were applied to the problems in biology, and his experiments had a large sampling size also. Larger sampling size, then errors will be minimized and the data will be more accurate. Data will be more accurate. That is in the case of the first he will do this mathematical logic and statistical analysis in biological sciences. Biological science, that is very important, and that is the major problems in biology. He will do these things, mathematical logic and statistical analysis he will apply. And his experiments are a large sampling size which have a great credibility. That data will be more accurate that he collected. Also, the confirmation of his inferences from experiments on successive generation of his test plans prove that his results point to the general rules of inheritance. That is the general rules of inheritance rather than being unsubstantiated ideas. But that is very important thing that is connected with Grigor Johann Mendel and his work. It is very important, and his experimental material was garden pea. Garden pea is called Pisum sativum. Garden pea is called Pisum sativum. Garden pea is experimental material, or the experimental plant. It is called a garden pea plant. That is called Pisum sativum. Okay, that were manifested as two opposing traits. There are two traits that will be taken in this each character. Example: the tall or dwarf plants. Tall or dwarf plants. In the case of the character stem height, the two opposing traits, it will be tall or dwarf plants. The next one is yellow or green seeds. Yellow or green seeds. Okay, this allow him to set up a basic framework of rules governing inheritance. Okay, which was expanded on by later scientists to account the diverse natural observation and the complexity inherent in them. That is the basic study that is done by Grigor Johann Mendel. Then it was expanded later by scientists. Later it will be expanded to the diverse natural observation, and it is very good contribution of this Grigor Johann Mendel. That also very important. And what are the experiments he conducted? That is the next part. Mendel conducted such an artificial pollination or cross pollination. Artificial pollination or cross pollination. Artificial hybridization. Artificial hybridization or cross pollination experiments using several true breeding pea lines. Several true breeding pea. Pea means garden pea. Garden pea. True breeding pea lines. And that is what is meant by true breeding. What is meant by true breeding pea lines or true breeding line? A true breeding line is that having undergone continuous self-pollination. A true breeding line is one that it is continuous. It has undergone continuous self-pollination and shows stable trait inheritance. Stable trait inheritance. There will be no variation. Stable trait inheritance and expression for several generations. That traits without any change. It will express for several generations. That is a true breed, or it is also called a pure breed. It is called a true breed, or sometimes it is also called a pure breed. Okay, true breed or pure breed. Yes, true breeding line or pure breed. It is called a pure breed. Okay. Then Mendel selected 14 true breeding pea plant varieties. How many pea plant varieties are selected by true breeding varieties are selected by Mendel in garden pea? There will be 14 true breeding pea plant varieties. That data is very important, as far as which were similar except for one character with the contrasting traits. contrasting traits. Okay. Some of the contrasting traits selected were smooth or wrinkled seeds, yellow or green seeds, inflated. Inflated means what? Full. Inflated means full, full or constricted. Constricted, then yellow pods and tall or dwarf plants. That are the some contrasting traits with each character. One character with the contrasting trait. Okay, but there are 7 characters he studied and with the 2 opposing traits. That is contrasting traits. That will be the case of it is given that 7 pairs of the contrasting traits in pea plant. Traits in pea plant, and that is studied by Mendel. That is also very important. That is 7 characters and its contrasting traits. That will be given in figure also and also in box. Each the character, seed shape, seed color, flower color, pod shape, pod color, then flower position and stem height. These are the 7 characters. 7 characters and its contrasting traits. You have already covered round, wrinkled, yellow, green, violet, white, full, constricted, green, yellow, then axile, terminal, and tall and dwarf. Tall and dwarf. Okay, these are the 7 characters and its contrasting traits. It is given either in this figure and also in next page it is given in a table. Next page it is given in the table. The contrasting traits studied by Mendel in P. Here also the 7 characters, its contrasting traits, that is dominant trait and a recessive trait. That is very important. That is the 7 characters. That will be asked several times. That dominant character studied in Garden P by Greer, Johann, Mendel. Any one. There will be 7 dominant features you have to study. That is in the case of the flower color, it will be violet, and seed color, it will be yellow. Pod color is green. Then seed shape, it will be round, and that is pod shape, it will be full. Full or inflated. It is called full or inflated. Inflated, full or inflated, and other one is constricted, recessive. Then next pod color, here it will be green. Then the flower position will be axile. Axile is dominant, and next one the stem height will be in the case of tall is dominant. But these are the dominant characters, and others are recessive characters. Okay. Then next we go through this. Mendel conducted is artificial pollination. Experiments in this garden bee. It is also given in the diagram also. That process, artificial hybridization process, the removal of anthers before the stamen dehisces. The anthers breaks or emasculation, removal of anthers, then transfer of pollen. All these process that will be for artificial hybridization. And his first cross you have already studied, that is called that is in the case of inheritance of one gene. One gene inheritance, or it is called monohybrid cross. That cross is called one character with the 2 contrasting traits studied at a time. That is called one gene interaction, inheritance of one gene. Let us take the example of one such hybridization experiment carried out by Mendel, where he crossed tall and dwarf. His first experiment that he will crossed this tall and dwarf pea plants to study the inheritance of one gene. Inheritance of one gene. And he collected the seeds. He collected the seeds produced as a result of this cross and grew them into generate the first hybrid generation. First hybrid generation. It is called F1 generation, or it is also called filial 1 generation. F1 generation, first filial generation. All this called. This generation is also called the filial 1 progeny or the F1 progeny. Then Mendel observed that all the F1 progeny plants were tall, like one of the parents. One of the parents, that will be, he will took the tall parent and the dwarf parent. Both the true breeding line, one is tall and the other one is dwarf. Both the parents. And here the F1, Mendel observed that all the F1 progeny plants were tall, like one of the parent. That is in the case of tall parent. None were dwarf in the F1 generation or first filial generation. Then he made similar observation for the other pairs of traits. He first do this first character, that is called stem height, then he repeat in other character with its contrasting traits, and also they will give the same result. That is the case of they will F1 also shows only one of the parental character, not other parent is not obtained in the F1 generation. F1 generation only one of the parental character is obtained. Then he found that F1 always resemble either one of the parents, and that the trait of the other parent was not seen in them. But he took the parents tall and dwarf plants, tall plant and dwarf plant, and after causing the F1 hybridization, as hybridization experiment is completed, then the seeds will be produced and it will be planted and it will produce the plants that are all tall. All the plants are tall, none of them were dwarf. Okay, then what happened? Next step, what will you do? Mendel then self-pollinated the tall F1 plants and to his surprise to obtain the F2 generation, next generation. He will self-pollination. The tall F1 plants to his surprise found that in the filial 2 generation or F2 generation, some of the offsprings were dwarf character. Some of the offsprings were dwarf. That was not seen, that character that was not seen in the F1 generation was now expressed. But F1 there will be no dwarf one, in the F2 the dwarf ones are expressed. The proportion of these plants that were dwarf were 1/4 of the plants, while 3/4 of the F2 plants were tall. Here in the case of this F2 generation, the proportion of the plants that were dwarf were 1/4. Out of 4, only 1. And in the case of 3/4 of the F2 plants were tall. That is the ratio, the tall and the dwarf. That will be in the ratio 3:1. 3 tall is to 1 dwarf. He will get in the F2 ratio. The tall and dwarf traits were identical to their parental type. That is very important. The tall and dwarf traits were identical to their parental type. Parental type means parental tall and parental dwarf. And did not show any blending. Did not show any blending or any mixing. Any blending or mixing. That is, all of the offspring were either tall or dwarf. All of the that offspring produced will be either tall or dwarf. None were of in between height, or there will be no intermediate height or in between tall and dwarf. That means there will be no blending of characters or mixing of characters that will take place in the F2 generation. There will be no blending of the characters takes place in the F2 generation. That is very important point. Okay. Then next, similar results were obtained within the other traits that he studied. That is in the case of, he first took this that is true tall and dwarf plants. Then next he repeated these experiments in other traits also, other characters with other traits, and he will give the same result. Similar results were obtained with the other traits that he studied. Only one of the parental trait was expressed in the F1 generation. That is the result. While at the F2 stage, both the traits are expressed in the proportion of 3:1 ratio. That is the phenotypic ratio. Phenotypic ratio. Phenotypic F2 phenotypic ratio. Monohybrid F2 phenotypic ratio, that will be 3:1. And the contrasting traits did not show any blending at either F1 or F2. Either F1, all of them are tall. All of them are tall, and it is similar to one of the parental tall. Parental genotype. And the second one, parental phenotype. Next, the contrasting test did not show any blending at either F1 or F2 also. F2 also they will get the both tall and dwarf ones also, but they will be same as similar phenotypic expression of the both the parental types. None were in between them, and there will be no blending of characters or mixing of characters. That is about the monohybrid cross. Monohybrid cross, or that is in the case of that is one gene interaction or one gene. Study a single gene interaction. Okay. Then what will be the results? Result he published, it will be, there will be where this the parental generation, that is tall and dwarf, pure tall and pure dwarf. True bits. Then F1 generation, all of them are tall. That is pictorially, diagrammatically representation of a monohybrid cross. We already explained in theory, it will be given in the diagrammatic representation. Tall and dwarf, then F1 generation, all of them are tall. Then self-pollination between, selfing means self-pollination between both the tall plants in the F1, and in surprise, the F2 there will be dwarf is appeared. Dwarf ones are appeared. In the F1 there will be no dwarf, F2 the dwarf ones are appeared. And that will be in the ratio 1/4. 1/4 of the F2, and 3/4 will be tall. This tall and dwarf will be the same as the phenotypic expression of the parental type. Okay, then what is the result he concludes? Based on this observation, Mendel proposed that something was being stably passed down from unchanged from parent to offspring through the gametes, over successive generation. And what we call that something is called, these things are called as factors. It is called a factors. According to Mendel, that substances that passing from one generation to next generation through that substances, and that substances or something, it is called factors. Now we call them as genes. The term genes was coined by Johansen, a scientist in the 1902. Johansen. Genes, therefore, are the units of inheritance. Genes, what we call the units of inheritance, and they contain the information that is required to express a particular trait in an organism. What gene will do? Gene will express a particular trait in an organism. That you already know. Okay, then genes which code for a pair of contrasting traits are known as alleles. That is very important term, alleles. What is alleles? Genes which code for a pair of contrasting traits. A pair of contrasting traits, that's called alleles. Or otherwise it is called what? That is, they are slightly different forms of the same gene. It is called slightly different forms of the same gene. Alleles are called slightly different forms of a gene. Or it is called genes which code for a pair of contrasting traits are known as alleles. And a gene is said to be an allelomorphic pair. A gene is said to be an allelomorphic pair. Okay. Then if you use alphabetical symbols for each gene, then the capital letter is used for the trait expressed in the F1 stage. That is alphabet. That is in the case of capital letter, capital T for tall, and a smaller alphabet for the other trait, that is called a smaller one, that is called a recessive, dwarf. So capital T is used for tall and small t for the dwarf, and capital T and small t are alleles of each other. Alleles of each other. Okay. Hence in plants, the pair of alleles for height would be capital T, capital T, small t, and small t, small t. About in the plants, the pair of alleles for height, it may be either capital T, capital T, small t, or small t, small t. Mendel also proposed that in a true breeding, tall or dwarf P variety, the allelic pair of genes for height are identical or homozygous. Allelic pair of genes for height are identical or homozygous. That is called capital T, and small t, small t. But that identical alleles will come in a gene, it is called a homozygous. Either it will be capital T, that is a homozygous tall, and small t, that is called that is a recessive. That is the in the case of dwarf. Capital T, and small t are called the genotype of the plant. What we called capital T, and small t, that is genetic constitution. Genetic constitution of the organism is called genotype of the plant, genotype. And the physical appearance or descriptive terms like tall and dwarf. Tall and dwarf means what? That is the physical appearance. Either it will be tall or it will be dwarf. That will be physical expression or physical appearance. It is called a phenotype. So what is genotype? What is phenotype? Genotype is the genetic constitution. Genetic constitution. Capital T, or small t, etc. Capital R, small r. All these things connected with the genotypes, and the descriptive terms like tall and dwarf, that is the physical structure or physical appearance. They are called a phenotype. Then what then would be the phenotype of a plant? That had a genotype capital T, small t. What is the phenotype of genotype capital T, small t? It will be tall. Capital T, small t, it will be tall. Okay. As Mendel found that the phenotype of the F1 heterozygote capital T, small t to be exactly like the capital T parent in appearance. Both are. As Mendel found the phenotype of the heterozygote capital T, small t to be exactly like the capital T parent in the appearance. That is same height. He proposed that in a pair of dissimilar factors, one dominates other. One dominates as in the F1, capital T and small t, and hence is called the dominant factor, while the other factor is called a recessive. In the case of dissimilar alleles come in a gene or factor, one will dominate the other. One will dominate the other in the F1, as in the F1. That you see in the capital T, small t, capital T with the small t, then the F1s are all capital T, small t. Here, the pair of dissimilar alleles like a capital T, small t factors, one dominates the other, and hence is called the dominant factor, while the other factor is called the small t is called recessive. In this case, capital T for tallness is dominant over small t for dwarfness, that is recessive. Okay. Then he observed identical behavior of all the characters or trait or pairs are he studied. Then next we use the symbols, gene symboling. Symboling is also very important, gene symbols, or it is called gene notation. Gene notation. Generally we call it is a convenient type, or it is a logically or convenient to use capital and lowercase of an alphabetical symbol to remember this concept of dominance. But dominance express in a symbol or express in other, we use it is convenient and logical to use the capital letter. and lowercase letter for the recessive character. Do not use capital T for tall and small d for dwarf, because you will find it difficult to remember whether capital T and small d are alleles of the same gene or character. But that is a mispractice, or it is a wrong practice. Capital D or small d, it is a wrong practice. Either capital T or small t, capital T or small t. Otherwise, there will be another gene notation that will be in the case of stem height. The recessive character will be dwarf, and the first letter of the dwarf, that is called first letter will be small d, and that will be taken in the capital. That will be taken as the capital D for tall. Capital D for tall and small d for dwarf. That type of notation is also applied. That will be also applied. That is capital D for tall and small d for dwarf. That is another type of gene notation or genetic notation. Gene symboling, another notation. That is very not very important. As you study in one case, that is in the case of your textbook, it is given the test cross. You study the test cross. That is a violet flower color. Violet flower color. It will be either the parent unknown genotype will be either capital W or capital W, small w. Homozygous dominant or heterozygous dominant. And in that book, in that cross, it is given the violet flower color is given the capital. Violet is a dominant character and it is given capital W. That is another method. That is in the case of the recessive character will be white. And the first letter of the white, that is W. That will be taken as capital case letter for violet flower. For violet flower, and small w for white flower. That is another type of gene notation. Okay. Capital T and small d are alleles. It is not alleles of the same gene or not. It will be no idea. So this practice is very wrong practice. Either capital T or small t. Capital T or small d is given. Okay. Then next, alleles. Alleles can be similar as in the case of homozygotes. Homozygous condition means what? Capital T, similar alleles. Similar alleles as in the case of homozygous, similar. Capital T and small t. Or can be dissimilar as in the case of heterozygote, that is capital T, small t. Okay. Since the capital T, small t plant is heterozygous for genes controlling one character, or it is a monohybrid. It is a monohybrid cross, and the cross between capital T and small t is called monohybrid cross. But capital T, small t plant is a heterozygous for genes controlling one character, that's called height. It is a monohybrid. It is a monohybrid, and the cross between capital T to small t and small t is a monohybrid cross. That is a typical monohybrid cross. And that experiment, that monohybrid cross, it is given first in diagrammatic. Here it is also given this monohybrid cross with the help of Punnett square. That square that we called Punnett square or checkerboard. A typical monohybrid cross conducted by Mendel between true breeding tall plants and true breeding dwarf plants. So that is another way it is to represent this monohybrid cross in Punnett square. Okay, that we study later. Okay, from the observation that recessive parental trace is expressed without any blending in the F2 generation, we can infer that when the tall and dwarf plant produce gametes by the process of meiosis. At the time of gamete formation, the meiosis will take place. And the alleles of the parental pair, that is either capital T or small t. Parental pairs are either capital T or small t. But here parental pairs will separate. They will be separate. Capital T and small t. They will segregate, or it is called segregate or separate from each other. And only one allele, either capital T, here also capital T, here small t or small t. One allele is transmitted to a gamete. Here capital T to one gamete and capital T to one gamete, and small t to one gamete and small t to one gamete. Only one gamete will move to, one allele will move to the gamete. Okay. This segregation of alleles is a random process. It is a random process. Okay, and so there is a 50% chance of a gamete containing either allele, or 50% of the gametes having containing the either allele, as has been verified by the results of the crossing. Okay, in this way, the gametes of the tall plants, capital T plants have the allele capital T, and the gametes of the dwarf plants, the small t plants have the allele small t. During fertilization, the two alleles, capital T from one parent, say through the pollen, it's either male or it will be female, and small t from other parent, then through the egg, they are united to produce zygotes. But that you have one capital T allele and one small t allele. The zygotes that you have one capital T allele and one small t allele. That is capital T with small t. This is zygote. To form zygote. Capital T to small t, capital T to small t, capital T to small t. That is in the case of during fertilization, the two alleles, capital T from one parent, say through the pollen. These are males and producing the pollens, and other one female that will produce eggs through the pollen, and small t from the other parent, then through the egg, through the eggs. These are eggs and these are the pollens, are united to produce zygote that have one capital T allele and one small t allele. In other words, the hybrids have capital T, small t. The hybrids have, the F1 hybrids are all have capital T, small t. Okay. Since these hybrids contain alleles which express contrasting traits, the plants are heterozygous. The plants are heterozygous. Okay. The production of gametes by that is very important. The production of the gametes by the parents and the formation of the zygotes in the F1 and F2 plants can be understood from a diagram called Punnett square. This square is called Punnett square. What do we obtain the Punnett square? What do we obtain the Punnett square? That is the production of gametes. The production of gametes by the parents. The formation of the zygotes. Formation of the zygotes in the F1 and in F2. F1 and F2. That is in the case of the production of gametes from parents and also the formation of the zygote during F1 generation and also in F2 generation. That can be understood from a diagram. It is called a Punnett square. That diagrammatic representation is called a Punnett square, and it was first Punnett square was first reported by there is a British genetist called Reginald C. Punnett. It was first developed by a British genetist, and he is called Reginald C. Punnett, R.C. Punnett. Reginald C. Punnett or R.C. Punnett. It is a graphical representation. It is a graphical representation to calculate the probability of all possible genotypes of the offspring, in a genetic cross. That is the main aim of this Punnett square or checkerboard concept. Graphical representation to calculate the probability of all the possible genotypes, of offspring in a genetic cross. And how we represent this Punnett square, or we represent the gametes in which part of this Punnett square, or the zygotes or offspring square which is represented. These will be given that next sentence. That is the possible gametes. The possible gametes are written on two sides. The possible gametes are written on two sides. What are the sides? That is at the top row and the left column. This is the top row and this one is the left column. Top row and left column. Top row and left column. Top row and left column. But that is very important. The possible gametes are written on two sides. Usually the top row and left columns. And all possible combinations or zygotes after crossing, after fusing, fertilization, the zygotes are formed. Combinations are represented in the boxes below. In the squares, which generates a square output. We get a square output. Our gametes are placed on either side of the square. That is in the case of top row and left column, and the products will be obtained through this square output form. Okay, and that's called Punnett square. That is very important. Punnett square. British genetist. It is developed by British genetist. That is called Reginald C. Punnett. R.C. Punnett. Okay. The Punnett square shows that parental capital T male and dwarf small t female plants, the gametes produced by them and the F1, that will be capital T small t progeny. Capital T small t progeny. That is in the case of F1. F1 will produce capital T small t progeny. The F1 plants of the genotype capital T small t are selfing. are self-pollinated, and the symbols are given for female and male. Here it is given female and this one is given as male. Are used to denote the female and males. Female males used to denote the female X and male pollen of the F1 generation. Okay. Then F1 plant of the genotype capital T small t when self-pollinated produces gametes. What are the gametes? Capital T and small t. And here is the other parent. It produces capital T and small t. It will produce produce gametes of the genotype capital T and small t in equal proportion, 50-50, equal proportion. Capital T and small t in 50-50. That is equal proportion. When fertilization takes place, when fertilization or crossing of the gametes, fertilization or fusion of the gametes, the pollen grains of genotype T, genotype is considered as male or other considered female. Okay, male or this is female, this one is male. But that is in the case of male T. That is in the case of male T. When the fertilizer, the pollen grains of genotype capital T have a 50% chance, pollen grains of genotype capital T have a 50% chance to pollinate X of the genotype capital T to capital T, that is 50%, as well as that of small t, about 50-50. Capital T to capital T to small t, that is chance will be 50-50. Okay, 50-50. As the pollen grains of genotype small t, pollen grains of small t mark. Okay, these are the pollens. These are the pollens and these are X. Female X. Okay, the pollen grains of the genotype capital T and capital T with the small t and as well as the genotype small t, pollinate X of the genotype as well as pollinate. Also pollen grains of small t have a 50% chance to capital T and small t. There will be capital T, small t, again capital T, small t, then next small t. Also the pollen grains of genotype small t have 50% chance of pollinating X of genotype capital T as well as of small t, as well as of the X of small t. About 50-50 chance on 50, the fusion of this male gamete to female gamete, the chance will be 50-50 as well as of the genotype small t. As a result of random fertilization, the resultant zygotes can be of the genotype capital T, small t or small t. Okay. About that, the genotypes obtained during this random fertilization, the resultant zygotes have either capital T, small t or small t. Then from the Punnett square, we have to analyze. It is seen that easily said 1/4 of the random fertilization lead to capital T, 1/2 lead to capital T, small t, and 1/4 to small t. That is called genotypic ratio. Genotypic ratio, that is 1/2 will be capital T, and 1/4 will be the capital T, 1/2 will be capital T, small t, and 1/4 will be small t. Though the F1 have a genotype capital T, small t, but the phenotypic character is tall. It will be tall. At F1, it will be all of them are tall. At F2, 3/4 of the plants are tall, where some of them are capital T, while others are capital T, small t. About the 3/4 of the tall plants, it is genotypically we separate it into capital T. Some of them are capital T, while others are capital T, small t. And these two are not externally, capital T and capital T, small t. Both are not externally easy to, or it is not possible to distinguish between the plants with the genotypes capital T and capital T, small t. Externally, we distinguish whether the genotype will be either capital T or capital T, small t. Both are tall. Tall, but it is not externally distinguished. Okay, then hence within the genotype pair, capital T, small t, only one character that is called capital T or tall is expressed. As the character capital T or tall is said to be dominate over the other allele called small t or dwarf character. It is thus due to this dominance of one character over the other, all the F1s are tall. That is law of dominance. Though the genotype is capital T, small t. And in the F2, what happens? 3/4 of the plants were tall, though the ratio, genotypically it will be 1/2 will be capital T, small t, and 1/4 will be capital T. 3/4 of the talls phenotypically 3/4 talls 3/4 tall ഇതാണ് phenotype 3/4 tall. ഇതിനെ നമുക്ക് genotypically എങ്ങനെ എഴുതാം? Genotype of 3/4 4 tall. That will be 1 by 2. 1 by 2 will be capital T small t plus 1 by 4 is capital T capital T. 3 by 4 tall. 1 by 2 will be capital T small t and 1 by 4 is capital T capital T. That is in the case of the 3 by 4 tall. 3 by 4 tall of the plants. Okay. This leads to the phenotype ratio of 3 by 4 tall is to 1 by 4 small t small t. But 3 by 4 tall 1 by 4 capital T plus 1 by 2 capital T small t, and next 1 by 4 is the small t small t. But 3 by 4 is tall and 1 by 4 will be small t small t. 3 by 4 tall will be either 1 by 4 capital T and 1 by 2 capital T small t in a 3:1 ratio. 3:1 ratio. But a genotypic ratio, 3:1, that is phenotypic ratio, that is 3 tall is to 1 dwarf. But ratio, genotypic ratio, it will be 1:2:1. 1 capital T, 2 capital T small t, and 1 is small t small t. That is the ratio, that is 1:2:1 genotypic ratio. Monohybrid genotypic ratio and 3:1 is the monohybrid phenotypic ratio. Genotypic ratio and phenotypic ratio. Okay, the next part that will be the 1 by 4 is to 1 by 2 is to 1 by 4 ratio. 1 by 4 is to 1 by 2 is to 1 by 4 ratio of what? The genotypes capital T, 1 by 4 capital T, 1 by 2 capital T small t, and 1 by 4 small t small t. This ratio it will be mathematically condensable. Mathematically condensable to the form of binomial expression. X plus by all square. Binomial expression condense mathematically. The 1 by 4 is to 1 by 2 is to 1 by 4 of the ratio of genotype capital T small t small t will be mathematically condensed to ax plus by all square in a binomial expression. What is the condition here? That is the gametes bearing genes. The gametes bearing genes capital T or small t in a equal frequency of 1 by 2. Equal frequency of 1 by 2. That is very important. Then the expression is expanded as given below. Over here, ax plus by. There a and b are constant and x and y are variables. A and b, that is 1 by 2 each. Frequency of each gametes, capital T and small t will be in 1 by 2. Our a and b are constant and x and y are variables. And what are the variables? They are the both the alleles, capital T and small t. That is 1 by 2 capital T plus 1 by 2 small t all squared. That is the binomial expression. It will be condensed. This theory will be, or this ratio will be condensed. This ratio will be condensed to ax plus by all square. That is 1 by 2 capital T plus 1 by 2 small t all square. That is the binomial expression. That will be condensed to binomial ax plus by all square. X plus by all square. That is the gametes bearing genes capital T or T in equal frequency of 1 by 2. Equal frequency of 1 by 2. The expression is expanded. When you expand the binomial expression expanded 1 by 2 capital T plus 1 by 2 small t all square is equal to 1 by 2 expand 1 by 2 capital T plus 1 by 2 small t into 1 by 2 capital T plus 1 by 2 small t. Expansion. That expand 1 by 4 capital T plus 1 by 2 capital T small t plus 1 by 4 small t small t. That is the binomial expansion. Binomial expression expansion. Expansion of the binomial expression. Binomial expression expand. Okay. Then next, Mendel self-pollinated the F2 plants and found that the dwarf F2 plants continue to generate dwarf plants in the F3 and F4 generation. That is next process. F3, F4 up to F2 F1 and F2 F3, F4 Next, from the preceding paragraphs, it is clear that though the genotypic ratio can be calculated using the mathematical probability by simply looking at the phenotype of a dominant trait, it is not possible to know the genotypic composition. The same problem. By simply looking the phenotype of a dominant trait, it is not possible to know the genotypic composition. Genotype it is an unknown genotype. Phenotypically both are tall. Both are tall. That is the case of tall plant from F1 or F2 as capital T or capital T small t in composition and cannot be predicted. Therefore, to determine the genotype of a tall plant at F2, Mendel crossed the tall plant from the F2 with the dwarf plant. Tall plant from the F2 with a dwarf plant, and that tall plant from the F2, it is unknown genotype. Either it may be capital T or capital T small t. It is crossed with the dwarf plant, and this cross is called test cross. This cross is called a test cross. In a typical test cross, an organism, here the pea plants here, showing a dominant phenotype and whose genotype is to be determined. Dominant phenotype, that will be tall phenotype, and the genotype is to be determined, is crossed with the recessive parent. Instead of self-crossing. Instead of self-crossing, it is crossed with the recessive parent. It is crossed with the recessive parent. And the progenies of such cross can easily be analyzed. The progenies, a cross conducted are progenies. Progenies analyze it, predict the genotype of the test organism or unknown organism. Unknown genotype. That is the progenies of such cross. That is the case of the F2 hybrid. F2 with the dwarf plant. F2 with the or showing a dominant phenotype. That genotype is to be determined. Is crossed with the recessive parent instead of self-crossing. The progenies of such cross can be easily be analyzed to predict the genotype of the test organism. Genotype of the that is called monohybrid test cross. It is called a monohybrid test cross. But that is F2 hybrid is cross back with the recessive parent. Cross back with the recessive, back cross with the recessive parent, and it is called a test cross. And the results of the test cross can be easily analyzed to predict the genotype of the test organism. Figure 5.5 shows the results of typical test cross where violet flower color, violet color flower is dominant over white flower color. That is given in the figure. It is given this diagram. Okay, this is the diagram of this test cross, diagrammatic representation of a test cross. This is the dominant phenotype. It is unknown. Genotype is unknown. Either it will be capital W or capital W small w. That is unknown genotype. That will be unknown. Okay, dominant phenotype, that will be unknown. Okay, the genotype is unknown. Then it is crossed back with the recessive parent. Homozygous recessive. Small w. It is crossed with the either recessive parent. Recessive parent cross imbo. What is the result? After obtaining the result analyze all if all flowers are violet and the unknown genotype is capital W. The result of this test cross, all the flowers are violet, then unknown flower is homozygous dominant, that is capital W. If the result of this test cross, the half of the flowers are violet and half of the flowers are white, the unknown flower is heterozygous, that is capital W small w. Capital W small w. That is in the case of the result. That is very important point. Here it is in the case of the progenies of such cross can easily be analyzed to predict the genotype of the test organism. That is, it is clearly shows all the offsprings that are showing this violet flower, that the unknown genotype is homozygous dominant, capital W, or half of the flowers are violet and half of the flowers are white, the unknown genotype is heterozygous, that is capital W small w. About that unknown genotype, either it will be capital W small w or capital W. Okay, then the monohybrid test cross ratio, monohybrid test cross ratio, that will be given 1:1. Monohybrid test cross ratio, that will be 1:1. Okay, based on his observation on monohybrid cross, Mendel proposed two general rules to consolidate the understanding of inheritance of monohybrid crosses. Today, these rules are called principles or laws of inheritance. First law is called law of dominance and second law is called law of segregation. That you already studied in this chapter. That is the law of dominance. It's very important. And second law, after completing the monohybrid cross, he established two rules or two principles of inheritance of monohybrid crosses. These rules are called principles or laws of inheritance. First law, law of dominance. What are the features of dominance? Law of dominance. Here in dominance, law of characters are controlled by discrete or discontinuous units called factors. Discrete or discontinuous units, that is alternate, this is distinct alternate forms, that is called discrete units are called factors, and factors are occur in pairs. In a dissimilar pair of factors, dissimilar, that is the both the alleles in the gene or alleles in the factors, one member of the pair dominant, and the other is recessive. That is called the law of dominance. But the law of dominance is used to explain, but the expression of only one of the parental characters in a monohybrid cross in the F1 and the expression of both in the F2. That is the law of dominance mainly used to explain the expression of only one of the parental characters in monohybrid cross in the F1. That is the case of tall. Capital T and small t, and the expression of both in the F2. It also explains the proportion of 3:1 obtained at the F2. 3:1 obtained at the F2. That is the law of dominance. Then law of segregation. What is law of segregation? It is based on the fact that alleles do not show any blending, that both the characters are recovered as such in the F2 generation. Though one of these is not seen in the F1 stage. That is very important. This law states that based on the fact that alleles do not show any blending or mixing, and then both the characters are recovered as in the F2 generation, though one of these is not seen at the F1 stage. Though the parents contain the two alleles during gamete formation, the factors or alleles of a pair segregate from each other such that a gamete receives only one of the two factors. That is called the law of segregation. Law of segregation. The parent contains two alleles during gamete formation. The factors or alleles of a pair segregate or separate. Segregate means separate from each other such that the gamete receives only one of the two factors. That is called the law of segregation, or it is sometimes called the law of purity of gametes. Law of purity of gametes. And it is the universally accepted law. It is the universally accepted law of Mendel. That is called the law of segregation, because in all sexually reproducing organisms at the time of meiosis, both the alleles will segregate and move to separate gametes. That is called the law of segregation, or it is called the law of purity of gametes. Though the parents contain two alleles during gamete formation, the factors or alleles of a pair segregate or separate from each other, and such that a gamete receives only one of the two factors. Gametes obtain only one of the two factors. Of course, a homozygous parent produces all gametes that are similar. That is capital T. It produces all the gametes are similar. That is capital T and capital T. While a heterozygous one produces two kinds of gametes. That is capital T, small t. Capital T, small t. It produces two types of gametes, capital T and a small t. Homozygous produces only one type of gamete, that is in the case of capital T. That are similar, while a heterozygous one produces two kinds of gametes, each having one allele with the equal proportion. Okay, then next we have to study that another one, that is law of segregation and law of dominance. Next we go through some post-Mendelian studies or post-Mendelian discoveries. It is connected with the other scientists. One such a interaction you have studied, that will be incomplete dominance. What is meant by incomplete dominance? That is in the case of here in the case experiments on peas were repeated using other traits in other plants. It was found that sometimes the F1 had a phenotype that did not resemble either of the two parents. And was in between two. But there will be, in nature, apparently there will be some variations are seen. In the case of Mendel, there will be no blending of characters or mixing of characters. He never get in this studies in 7 characters studied in garden pea or Pisum sativum. When the experiments conducted on peas were repeated using other traits in other plants, it was found that sometimes the F1 had a phenotype. That did not resemble either of the two parents. Okay, and that did not resemble either of the two parents and was in between two. രണ്ടിന്റെ ഇടയിൽ വരും, in between two. Okay. അങ്ങനെ വരുമ്പോൾ നമ്മൾ അതിനെ എന്തിനെ ഒബ്ജക്ഷൻ ആയി, that is complete dominance or Mendelian അല്ലെ dominance, law of dominance against I, that's called incomplete dominance. Did not resemble either of the two parents and was in between the two. Between the two. A typical example ആണ് ഇന്ന് നമ്മൾ പറയുന്നത്, that's called incomplete dominance or partial dominance. അല്ലെ the inheritance of a flower color in the dog flower. അല്ലെ dog flower. Dog flower എന്ന് വിളിക്കുന്ന പേരാണ് snapdragon or antirrhinum. Scientific name is antirrhinum. അല്ലെ dog flower is also called snapdragon, that's English name, and antirrhinum is a botanical name. Is a good example to understand incomplete dominance. So a good example of incomplete dominance. Incomplete. In a cross between incomplete dominance prove the parental generation, gametes, the first F1 generation, then F2 gametes, again a gametes, then F2 generation. That is simple monohybrid cross. That is a simple monohybrid cross, but that in the case of the F1 generation does not resemble either of the parents or in between two. Okay. Cross and these F1s are selfing. Self-pollination. After self-pollination, then F2 will obtain the ratio of that is 1:2:1 in the case of phenotypic ratio. That is typical monohybrid phenotypic ratio is 3:1 is changed. It will be changed to 1:2:1. That we explain here. That will be explained here in the textbook. Okay. The flower color in the dog flower, that is called snapdragon or antirrhinum, is a good example to understand incomplete dominance. It is also done in another plant called 4 o'clock plant, Mirabilis jalapa. Another plant, it is also done this incomplete dominance in Mirabilis jalapa. Mirabilis, 4 o'clock plant. 4 o'clock plant is also another example for understand this incomplete dominance. In a cross between true breeding red flowered and true breeding white flowered plants, capital R and small r, the F1, about the gametes it is obtained capital R, here it will be small r, and the F1 generation having not red, not white, neither red nor white, it will be all are pink in color. All are pink in color. That is the very important feature. Here the true breeding red flowered and true breeding white flowered plants, the F1 was pink and the F1 was self-pollinated. F1, that all the pink, capital R, small r is self-pollinated. F2 result in the following ratio. That is the ratio 1 red is to 2 pink is to 1 white. 1 red is to 2 pink is to 1 white. But that the monohybrid cross, that both genotypic and phenotypic ratio will be same. Both the genotypic and phenotypic ratio will be same. Here in the case of normal monohybrid phenotypic ratio is 3. So they will expect also. The Carl Correns will do this experiment, incomplete dominance. And he will also expect that ratio of monohybrid phenotypic ratio of 3:1. 3:1. But it will be changed to 1:2:1. That means 1 red is to 2 pink is to 1 white. And the genotypic ratio is also 1 capital R, 2 capital R, small r, and 1 small r. But both this genotypic ratio and the phenotypic ratio, both genotypic ratio and phenotypic ratio, both are same. Phenotypic and the genotypic ratio will be same. That is 1:2:1, 1:2:1. Okay, that is the very important data. Here it will be. Here the genotypic ratio are exactly as would expect in any Mendelian monohybrid cross. What is the ratio? Genotypic ratio exactly would expect in any Mendelian monohybrid cross. But the phenotypic ratio had changed from 3:1 dominant recessive ratio. That will be changed to 1:2:1. 1:2:1. Okay, then what happened? What will happen during this incomplete dominance? Here the capital R was not completely dominant over small r. But that is it is not completely dominant over small r. But there will be a partial dominance. There will be an incomplete dominance or partial dominance, or here the characters are get mixed, or the blending of the characters, blending of the characters. Either the red and white is mixed to form the pink color. Both these characters. That will be very important. it is possible to distinguish capital R, small r as pink from capital R and small r white. Capital R red and small r white. That is in the case of this distinguish capital R, small r as pink from capital R red and small r white. So that is another important cross that is called here the phenotypic ratio 1 red is to 2 pink is to 1 white, and the genotypic ratio is also 1 is to 2 is to 1. That is 1 capital R, 2 capital R small r and 1 small r. So that is the case of incomplete dominance. It is studied in snapdragon or dog flower. And another plant it is called Mirabilis delapa, 4 o'clock plant. Okay. The next one is the explanation of the concept of dominance. That you already studied explanation. Why some genes are showing dominant and why some are recessive. How do we explain this? The major things it is every gene, as you know, contains the information to express a particular trait. In a diploid organism, there are 2 copies of each gene as a pair of alleles. Now, these 2 alleles need not always be identical as in heterozygote. Heterozygote, there will be dissimilar alleles. 2 alleles will be need not always be identical. One of them may be different due to some changes that it has undergone, which that we will study in the next chapter, and which modifies the information that particular allele contains. But that is the modification of allele that will changes the phenotype. And which allele will act as the dominant allele? That is very important. Prove in the experimental that is a metabolic reaction. That will be a gene that will control the production of an enzyme. A gene that will produce, example, a gene that contains the information producing an enzyme. Now there are 2 copies of this gene. The 2 allelic forms. 2 copies, that is 2 allelic forms in that gene. Let us assume, as is more common, that the normal allele produces the normal enzyme. Normal allele, that means that normal gene having 2 alleles, that 2 allelic forms that will normal allele produce normal enzyme that is needed for the transformation of a substrate. But normal enzyme form enzyme will transform a substrate. Transformation of a substrate, that is the phenotype. Now phenotype enzyme action which phenotype explain enzyme action. Here it is the case of normal allele producers. Normal allele capital S normal allele. Genotype normal gene, normal S, normal capital S and capital S, normal gene. It will produce the normal enzyme. Normal enzyme, and that will convert substrate to products. Okay. Substrate to product. Okay. Then theoretically, the modified allele, the normal allele, normal alleles, capital S, normal allele, could be responsible for the, theoretically, the modification or modified allele could be responsible for the production of. In alleles modification, response mark, that is in the case of, it will be, the phenotype will be changed, or in the enzyme action will be affected. The condition is the first condition, that theoretically the modified allele will shows the production of normal or less efficient enzyme. That is the first condition. Normal or less efficient. Second, non-functional enzyme, or third, no enzyme at all. For first case, we compare first one, the normal and less efficient enzyme. What happens in the case of normal or less efficient? That is the phenotype. That means the enzyme production will takes place. Because the enzyme and there will be gene will working. The normal or less efficient enzyme. That means enzyme production will takes place, and it will be normal condition or slightly less efficient condition also. But here the action will takes place. In the first case, the modified allele is equivalent to the unmodified allele. Which allele is modified? That is in the case of the capital S and the small s. Capital S and the small s. This allele is modified. Capital S will be modified to small s. The modified allele is equivalent to the unmodified allele. Small s is equivalent to the unmodified allele. It will produce the same phenotype or trait. Means normal enzyme action or normal enzyme, production of normal enzyme that will result in the transformation of substrate S. Substrate S. Enzyme substrate to product. Substrate and product that will be the enzyme action. Here this modified allele will equivalent to unmodified. This one is unmodified allele. unmodified allele. That is the normal allele, unmodified allele. This one is the modified allele. So modified allele is equivalent to the unmodified allele. Unmodified allele, and it will produce the same phenotype or trait. That result in the transformation of substrate S. Okay, such equivalent allele pairs are very common. That means capital S and small s. That means one dominant allele and one recessive allele, and that is called the such allele pairs. Such allele pairs, that is called heterozygous. Such equivalent pairs are very common. For most of the population, we see this. Most of the heterozygous forms are most common. It will be, but if the allele produces a non-functional enzyme or no enzyme, it will produce second condition, non-functional enzyme or no enzyme. Third condition, no enzyme. The phenotype may be affected. The phenotype may be affected. That means the phenotype, that will be the enzyme action not take place or no product formation, that may be affected. Okay. The phenotype or trait will only be dependent on, the phenotype or trait will be only dependent on the functioning of the unmodified allele. Functioning of the phenotype depend in the area functioning of the unmodified allele. For unmodified allele dependent on phenotype. If modification there will be enzyme production, that enzyme will be non-functional. Here the unmodified or functioning allele, unmodified, or it is also called functioning. It is also called functioning allele, unmodified or functioning allele, which represents the original phenotype is the dominant allele and the modified allele is generally the recessive allele. Well, that is what is which represent unmodified allele, unmodified allele which represents the original phenotype, which will represent the original phenotype, and it is the dominant allele. It is called the dominant allele. Dominant allele, and the modified allele. Modified allele is called the recessive allele. It's called the recessive allele. It is called the recessive allele. And in the example about the recessive trait is seen due to non-functional enzyme or because no enzyme is produced. Recessive trait means smallest. The alleles will be smallest, recessive. But here it is in the case of capital smallest, that is unmodified allele will represent the phenotype, and it is called the dominant allele, and other one is called the recessive allele. The modified allele is called the recessive. But the phenotype is governed by the dominant allele, or it is called unmodified allele, or it is called a functioning allele. It is called a functioning allele. Okay. The unmodified or functioning allele, which represent the original phenotype, is the dominant. It is the dominant allele, and other one is called, generally the modified allele is called recessive, and it will produces the non-functional enzyme, because non-functional enzyme is not working. That is called non-functional, and it is the result same as the no enzyme is produced. No enzyme produced in non-functional enzyme, there will be no conversion of substrate to product. Enzyme work ചെയ്താലാണ് സബ്സ്ട്രേറ്റിനെ പ്രൊഡക്റ്റ് ആക്കാൻ പറ്റുള്ളൂ. അപ്പൊ നോൺ ഫങ്ഷണൽ എൻസൈം എന്ന് പറഞ്ഞാൽ അവിടെ സബ്സ്ട്രേറ്റിനെ എന്താക്കി മാറ്റുന്നില്ല, പ്രൊഡക്റ്റ് ആക്കി മാറ്റുന്നില്ല. അത് തന്നെയാണ് എന്ത് ചെയ്യുന്നത്, നോ എൻസൈമിന്റെ എഫക്ട്. Same effect that will be no enzyme. Up here the alleles will be the recessive, generally the recessive allele. About the recessive trait will be due to non-functional enzyme or no enzyme is produced. That is the concept of dominance. But it is explained in the enzyme production, that a gene is responsible for an enzyme. A gene is responsible for an enzyme production, not in the other characters like physical characters. It is clearly established this concept of dominance. That will clear. That will be clear. This the concept of dominance in this experiment. That is very important experiment. The next one is codominance. Codominance is another that is another post-Mendelian inheritance. Post-Mendelian inheritance. Till now we were discussing the crosses where the F1 resembled either of the two parents. Till now we were discussing the crosses where the F1 resembled either of the two parents. That is called dominance, or was in between, incomplete dominance. In between, resemble either of the parents. F1 resemble either of the parents, that is law of dominance, Mendelian. Or was in between, that is called incomplete. Red and white, there will be pink color, incomplete dominance. But in the case of codominance, the F1 generation resembles both the parents. Resembles both the parents. A good example is different types of RBC, red blood cells, that determine the ABO blood grouping in human beings. A typical example for this codominance, it is studied in ABO blood grouping. It is a typical example for this codominance studied in ABO blood grouping. ABO blood groups are controlled by the gene I. The gene is called I gene, isoagglutinogen. Agglutinogen. That's I full name. In short form, I. The plasma membrane of the RBC has the sugar polymers that protrude from the surface, and the kind of sugar is controlled by the gene. Our plasma membrane, our sugar polymers protrude from its surface, and it is a kind of sugar is controlled by the gene. I gene. The gene called I gene. The gene I has three alleles. What are they? IA, IB, and small I. Three alleles. And the alleles IA and IB will produce a slightly different form of sugar. IA produces a type of sugar, IB will produce slightly different form of sugar. While small I does not produce any sugar. Because humans are diploid organism. We are a diploid organism. To an organism. Each person possesses any two of the three I gene alleles. Any two of the three I gene. Three I gene alleles, IA, IB, and small I. About any two of the three I gene alleles. Okay. About we are diploid organism, and we are chromosome number is diploid to 1, and it is having only two copies of allele. Either of these three, IA, IB, and small I, any two of the three alleles, I gene alleles will be present in an individual. But that is very important. Individual have only two alleles. In the population, it will be three alleles. That is the human population. The blood grouping, ABO blood grouping is controlled by three alleles. In a population, human population. In a human individual has only two alleles. Okay. Then when IA and IB are completely dominant over small I, that means, in other words, when IA and small I are present, only IA expresses, because small I does not produce any sugar. Small I does not produce any sugar. IA produces one type of sugar, IB will produce other type of sugar, and small I does not produce any sugar. Okay. Here in the case of IA and small I are present, only IA expresses, because small I, in other words, it does not produce any sugar. Then next, when IB and small I are present, IB expresses. When IA and IB are present, together, they both express their own types of sugar. That is called codominance. Both the IA and IB are present together. Both will express their own sugars, their own type of sugars. It is called codominance. So in the F1, both will represent. IA and IB both will present their sugars or their products or their phenotypes. It will express. IA and IB are present together. It is called codominance. That is the AB blood group is a typical example for this codominance. IA and IB, and it is AB blood group is coming under ABO blood group system. Hence, red blood cells have both A and B types of sugars. Since there are three different alleles, IA, IB, and small I. There are 6 different combinations of these 3 alleles. These 3 alleles in the 6 combinations. 6 combinations that are possible, and therefore a total of 6 different genotypes of the human ABO types are present. 6 genotypes. You have studied that 6 genotypes. What are the 6 genotypes in the that will be given in the table 5.2, next table. And how many phenotypes are possible? How many genotypes are in the case of ABO blood group system? There are 6 genotypes that you study in the table also. 6 genotypes. 6 genotypes. What are the 6 genotypes? That is in the case of IA, then IA, small I, then IB, then IB, small I, then IA, IB, then small I. 1, 2, 3, 4, 5, 6. 6 genotypes. 6 genotypes, and next, how many phenotypes are possible? There are 4 phenotypes. What are the 4 phenotypes? 4 phenotypes are A, B, AB, and O. A group, B group, AB group, and O group. There are 4 phenotypes. 4 phenotypes, and 6th, 4 phenotypes. ABO blood group system consists of 4 phenotypes, 6 genotypes, 6 genotypes and 3 multiple alleles, 3 alleles. 3 alleles and 1 gene. 1 gene, that is I gene. 1 gene, 3 alleles, 6 genotypes, 4 phenotypes. And an individual have only 2 alleles. An individual have 2 alleles. In a population that will control 3 alleles. Okay. Then the table is also given. Blood grouping questions are showing the genetic basis of blood groups. Allele from parent 1, allele from parent 2, genotype of the offspring and blood type. Okay, IA, that is A blood group, blood type in A, then the genotype is IAA. Then IAIB, that is AB, IAIB, that is AB blood group. Then again IAIA, that is like a third genotype, that is also A blood group. Then IBIA, that is IAIB, that's same. That's same genotype that we already noted here. Then IBIB, that's another genotype. Then next IB small I, then also B group. Then small I, that will be O group. About here it is the case of 2 A group, 1 AB group, 2 B group, and 1 O group. There will be 6 genotype. But 7 genotype or AB is same. AB will be same. That's in the case of AB blood group. That's only 1 genotype. So there are 6 genotypes and 4 phenotypes. What are the 4 phenotypes? A, AB, B, O. A, B, AB, and O. 4 phenotypes. Okay. Then, do you realize the example of ABO blood grouping is also provides a good example for multiple alleles? And here you see that there are more than 2. There are 3 alleles governing the same character. Since an individual, only 2 alleles can be present. Very good. This is very important. 3 alleles governing the same character. Since in an individual, only 2 alleles can be present. Only 2 alleles can be present. Multiple alleles can be found only when population studies are made. That is a very important point. But this is called multiple alleles. Multiple alleles. In ABO blood groups is also good example for multiple. What is multiple alleles? There are more than 2 alleles. That means 3 alleles governing the same character, or 3 or more alleles. More than 2 means 3. 3 or more than alleles governing a same character. It is called multiple allelism. It is called multiple allelism. Since an individual, only 2 alleles can be present. 2 alleles can be present. Multiple alleles can be found only when population studies are made. Yes. Okay. Then next, another one, that is in the case of a single gene product may produce more than one effect. That is called pleiotropism. That is an example for another one that is called pleiotropism. A single gene will produce more than one effect. Single gene will produce more than one effect or more than one phenotype. That is called pleiotropism or manifold effects of a gene. Manifold effects of a gene. For example, starch synthesis in pea seeds is controlled by one gene. It has 2 alleles, capital B and a small b. Capital B and a small b. Then starch is synthesized effectively by capital B homozygous, and therefore large starch grains are produced. In contrast, small b homozygotes have lesser efficiency in starch synthesis and produce smaller starch grains. But capital B will produce large starch grains that will effectively synthesize of starch, and small b is lesser efficiency and it will produce only small starch grain. Then after this starch grain synthesis, the maturation of the seeds. After starch grain synthesis and maturation of the seeds, capital B seeds are round. Capital B, the seeds after maturation, the seed shape will be round shape, and small b seeds will be wrinkled. It will be wrinkled. Then heterozygotes, that is capital B, small b will also produce the round seeds. What happens? Here capital B seems to be the dominant allele. Like capital B round up, small b wrinkled, capital B small b, that will also shows the dominant. That will be capital B small b, it will be round, again round. So that capital B seems to be the dominant allele. But the starch grain that produced are of capital B small b will of intermediate size. So capital B will produce, the starch grain size will be large, small b will produce the small starch grains, and capital B, small b will produce intermediate size in the starch grain. Starch grain will be intermediate. Over here, if the starch grain size is considered as the phenotype, starch grain size is considered as the phenotype, then from this angle, the allele shows incomplete dominance. Whereas the seed shape is taken, that if seed shape is considered, then from that angle, that will be in the seed shape, it will be shows the law of dominance. It will show the complete dominance. Here, if starch grain is produced, it will be the incomplete dominance. About one gene product will shows different phenotypes or different effects. More than one effect. More than one. One is dominance, other one is called incomplete dominance. And it is also then example for that pleiotropism. Therefore, the dominance is not an autonomous feature of a gene or the product that has information for. It depends as much on the gene product and the production of a particular phenotype. From this product, it does the particular phenotype that we choose to examine. In the case of more than one phenotype is influenced by the same gene. That is called more than one phenotype is influenced by the same gene. That is called pleiotropism. More than one phenotype is influenced by the same gene. That is called manifold effects of the gene. Okay, then last part that in the first class we covered up to this inheritance of two genes. That is called dihybrid cross. There is only one gene interaction you studied, that's called monohybrid cross. Here it will be that is inheritance of two genes, that is studied in two characters studied at a time. And here the pea plant with the seed shape and seed color. He took the two characters, seed color and the seed shape. And seed color, that will be yellow color, and that is in the case of the color, it will be yellow and green. Seed dominant yellow color and green seed color, recessive green, and the round shape is dominant and wrinkled is recessive. Then Mendel found the resulting from the crossing of the parents had yellow colored and round shaped seeds. In the case of F1 generation, he got a round shaped and yellow color seeds you will obtain. And in the F2, that will be in the case of selfing this F1. The F2 will shows the ratio of 9:3:3:1. And the yellow color was dominant over green and the round shape dominant over wrinkled. These results were identical to those we made a separate monohybrid crosses between yellow and green seeded plants and between round and wrinkled seeded plants. So that is, this yellow color was dominant over green and round shape is dominant over wrinkled. These results were identical to those he got when he made separate monohybrid crosses between yellow and green and also round and wrinkled seeded plants. Okay, in the genotypic symbol, that is the genotypic symbols. Capital 5 for dominant yellow seed color and small 5 for recessive, and capital R for round seed shape and small r for wrinkled seed shape. The genotype of the parents can be written as capital R, capital Y for round and yellow, and small y for wrinkled and green. The cross between the two plants can be written down as in the figure. That is given 5 points. Figure the parents will be capital R, capital Y, small r, small y. That is round yellow and wrinkled green. Okay, that is the parents. Cross between these plants written in the showing the genotypes of the parent plants. Then the gametes obtained from these parents, capital R, capital Y, with the small r, small y. What are the parent gametes it's obtained? Capital R and capital Y. Here the gametes are small r and small y. That is the gametes. This parent generation, that one is the gametes. Okay, these are the gametes. Then showing the genotype of parent plants. The gametes capital R, capital Y and small r, small y unite to fertilization. They will fertilize to produce. On fertilize produce F1 hybrid. F1 hybrid is capital R, capital Y, small y. They all are round yellow. F1 are all showing round yellow. Law of dominance. Then next, what do you do? That is in the case of in the F2. That is when Mendel self-hybridized the F1 plants. He found that 3/4 of the F2 plants had yellow seeds and 1/4 had green. And the yellow and green color segregate in a ratio of 3:1. The color yellow and green seed color segregate in a ratio of 3:1, and round and wrinkled seed shape also segregated in a ratio of 3:1, just like a monohybrid cross. Well, this is two characters, round and wrinkled and yellow and green. that will be segregated in a ratio in the F2. In the F2, that is in the case of 3:1 each. 3:1 each. Then the result of this F2, F1 selfing of this F1, F2 will produce, that is in the case of phenotypic ratio 9 round yellow, 3 round green, and 3 wrinkled yellow, and 1 wrinkled green. Here it is in the case of the dihybrid phenotypic cross. Phenotypic ratio, dihybrid phenotypic ratio. Here the gametes are taken here. Gametes, it is obtained from this capital R, capital Y, small y. That we will study later. That is the law of independent assortment. We have to study during the meiosis, the gamete formation. Here are the two pair of characters. That is the case of two pair of set of characters, traits that found in a hybrid. That will segregate. That is capital R, and capital Y. That's two characters. That is seed shape and a seed color. These two pair of characters in a hybrid, that will segregate independently. They will segregate independent assortment. They will assorted independently, or independent segregation from capital R to small r and capital Y to small y. And that produces the 4, that is 50-50 chance for capital R to capital Y and capital R to small y, and small r to capital Y and small r to small y, and produces 4 types of gametes of each having equal frequency, 25% each, 25, 25. Each. That we will study later. That is in the case of the independent assortment, law of independent assortment. In the dihybrid cross, the phenotypes round and yellow, wrinkled and yellow, round green and wrinkled green appeared in the ratio 9:3:3:1. That is in the case of dihybrid F2 ratio, the phenotypic ratio. 9 round yellow, 3 wrinkled yellow, 3 round green, and 1 wrinkled green appeared in the ratio 9:3:2. This ratio was observed for several pairs of characters that Mendel studied. It will be studied in other characters also. Other characters, other pair of characters, set of characters, other set of characters. And the ratio of 9:3:3:1 can be derived as a combination series of 3 yellow:1 green, or with 3 round:1 wrinkled. That is the combination series of 2 monohybrid cross. What are they? That is in the case of seed color and seed shape. 3 yellow:1 green with a 3 round:1 wrinkle. It is a combination series, and this derivation can be written as follows. That is very simple. 3 round:1 wrinkle into 3 yellow:1 green. Then it will be 3 into 3, 3 into 3, that is 9 round yellow. Next 3 into, that is 1 into 3, 1 into 3, that is 3 wrinkled yellow. Then 3 round into, that is the case of 3 round into 1, that is the 3 round green, and 1 wrinkle into 1 green, that's called a wrinkled green. That is the ratio, that is combination series of 3 round:1 wrinkled into 3 yellow:1 green. That will be 9 round yellow, 3 wrinkled yellow, 3 round green, and 1 wrinkled green. Based upon such observation on dihybrid crosses between plants in 2 different traits, Mendel proposed a second set of generalization that we called Mendel's laws of independent assortment. Mendel's laws of independent assortment. That law states that when 2 pairs of traits are combined in a hybrid, the segregation of one pair of character is independent of the other pair of character. One pair of character is independent of the other pair. That is capital R, small r. Capital R, small r is independently segregate with the capital Y, small y. When the 2 pairs of traits, are combined in a hybrid, that is capital R, capital Y, small y. Segregation of one pair of character is independent of the other pair of character, like capital Y and small y. It will be independent. Okay, that is very important. Independent of the other pair of characters. That's called the law of independent assortment. Okay, the Punnett square can be effectively used to understand the independent segregation of the 2 pairs of genes. 2 pairs of genes during meiosis. Independent segregation of the 2 pairs of genes during meiosis, and the production of eggs and pollen in the F1 capital R, capital Y, small y. F1 parent, capital R, capital Y, small y parent. This parent, other parent, capital R, small r, capital Y, small y. Second parent. That is second parent. First parent and second parent. Two parent. Consider the segregation of one pair of gene, capital R and small r. 50% of the gametes, 50% of the gametes have the gene capital R. 50% of the gametes are having capital R and other 50% have small r. Other 50%. For 50% gametes are capital R and 50% gametes are small r. 50%. Now, besides each gamete having either capital R or small r, it should also have the allele, capital Y or small y. It should also have the allele capital Y or small y, capital Y or small y. The important things to remember here is that segregation of 50% of capital R and 50% of small r. 50% of capital R and 50% of small r is independent from the segregation of 50% of capital Y and small y, capital Y and small y, capital Y and small y. Therefore, 50% of the small r bearing gametes, small r bearing gametes as capital Y, capital Y, 50%, and other 50% have small y. Other 50% have small y. Our small r with the capital Y 50%, small r with the small y 50%. 50%. Okay. Then similarly, 50% of the capital R, 50% of the capital R bearing gametes with the capital Y, 50%. 50%. Capital R as capital Y and other 50% as small y. Other 50% as small y. Okay. But that is the case. Similarly, 50% of the capital R bearing gametes have capital Y and other 50% as small y. Thus, there are 4 genotypes of the gametes. 4 genotypes. What are they? First one, that is the case of capital R to capital Y. Capital R to capital Y. First gamete, capital R to capital Y. Then capital R to small y. Then small r to capital Y. Then small r to small y. There are 4 types of gametes. There are 4 types of gametes. Here it will be 50% thus there are 4 genotypes of the gametes or 4 types of pollen and 4 types of X. Repeat 4 types of X. 2 parents. The 4 types are capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. Each with a frequency of 25%. Each with a frequency of 25, 25, 25, 25. 25. All are having 25 or 1 by 4. Each are 1 by 4. Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. That is called the independent assortment during the gamete formation, during meiosis. Okay. Yes. Law of independent assortment with a frequency of 25% or 1 by 4th of the total gametes are produced. When you write down the 4 types of X and pollens on the 2 sides of a Punnett square, it is very easy to derive the composition of zygotes. That gives rise to the F2 plants. And all there are 16 squares. How many different types of genotypes and phenotypes are formed? Atra genotype, atra phenotype. That is the formula you have studied. 9:3:3:1, 4 phenotypes. And how many genotypes will be? There will be 9 genotypes. And the genotypic ratio is 1:2. Genotypic ratio, dihybrid genotypic 1:2, 2:2, and 4:1:2, and 1:2:1. 1:2:1. I think 1:2:1, 2:4:2, and 1:2:1. That is 9 genotypes. 9 genotypes. And 4 phenotypes. 4 phenotypes. 4 phenotypes is 9:3:3:1. 9 round yellow, 3 wrinkled yellow. 3 round green and 1 is wrinkled green. 1 is wrinkled green. That is about this dihybrid cross. Okay. But these are all things connected with this 2 gene interaction or dihybrid cross. And the data you explain this F2 ratio. F2 ratio in Punnett square. F2 ratio in the Punnett square, that is very important. You have to explain all these things. there will be two diagonal lines. It is given first diagonal line here. It will be given all the homozygous forms, and here it will be the completely true dihybrids. There will be here, and other monohybrids it will be given in 2:2:2:2. All these data it will be given in this dihybrid F2 ratio. Dihybrid F2 ratio. All this data will be very important. Our data is genotypic forms. These are the 4 genotypes. It will shows all of them are homozygous. All are homozygous. Capital R, capital R, capital Y, small r, capital Y, capital R, capital R, small y, small r, small y, small y. They are all the completely homozygous forms. Completely homozygous. And these are the true dihybrids. True dihybrids. Next diagonal, capital R, capital Y, small y, capital R, capital Y, small y, capital R, capital Y, small y, capital R, capital Y, small y. That is called the true dihybrids. And then one character will show hybrid. That is in the case of capital R, capital Y, capital Y, capital R, capital Y, capital Y. Another one, so that will be 2 monohybrid characters. Then next one, capital R, capital Y, small y, capital R, capital Y, small y. Then small r, capital Y, small r, capital Y, small y. Then next one, capital R, small y, small y, capital R, small y, small y. So that is a ratio number that is the case of 1:2:2, then 4:2:1, 1:2:1. That is based on 9 round yellow, 3 round green, and 3 wrinkled yellow, and 1 wrinkled green. Okay, but these things that will be very important in the case of this dihybrid cross. All these things we are identified, and how many types of gamete formation, how many gamete formations takes place during this dihybrid, how many genotypes are produced, how many phenotypes are produced. All these things are very important. Well, up to this part, this is our today's portion. That will be almost time is over. Then next class we continue with this chromosomal basis. That is called the chromosomal theory. It is connected with the chromosome theory. These are based on the genetic basis, or that is genic level. All this genic level we have covered. Next is in the case of next class onwards, we have to go through this chromosomal basis or chromosomal theory of inheritance. Chromosomal theory of inheritance, that will be you already studied in this chapter. Okay, Mendel published his inheritance of work in 1865. His studies will be, or his experiments will conduct up to 1856 to 1863, and his work is inheritance of characters is published in the year 1865. But for several reasons it will remain unrecognized till 1900. Firstly, the communication. What are the main drawbacks here? It will be firstly the communication was not easy. That is very important. That is not publicized, not well publicized. It is not widely publicized. That is the first reason it will be remain unrecognized. For 1900. Okay. Secondly, is concept of genes. Concept of genes or factors. In Mendel's words, it will be stable and discrete. It will be stable and discrete or discontinuous units. Discontinuous units that control the expression of traits and of the pair of alleles which did not blend with each other. Was not accepted by his contemporaries as an explanation for the apparently continuous variation seen in nature. That is, the blending of characters is not reported by Mendel. But in the contemporary scientists, at that time, they will be contemporaries, they will explain the apparently continuous variation seen in nature. And they will show that is in the case of the variation, or it will be blending of characters will be seen. Okay. Thirdly, the Mendel's approach of using mathematics to explain biological phenomena was totally new. And it is unacceptable to many of the biologists for his time. Finally, though Mendel's work suggested that factors or genes were discrete units, he could not provide any physical proof for the existence of factors or so what they are made up of. That is very important. These are the main reasons why, because these his works are till recognized 1900. Very important things. Then in 1900, the three scientists, De Vries, Correns, and von Stermark, Hugo De Vries, Carl Correns, and von Stermark, independently rediscovered Mendel's results on the inheritance of characters, and also by this time the advancements in microscopy, all these touches, or the scientists able to carefully observe the cell division, okay, inside the nucleus, The cell division is very clear. Structures inside the nucleus that appear to be double and divide. Inside the structures found inside the nucleus will be double and divide just before each cell division. These things are called chromosomes. That substances that will or structures in the nucleus appear to be double and divide before each cell division. These are called chromosomes. Chromosomes, colored bodies. Chroma means colored, somes means bodies. Soma. As they were visualized by staining. By 1902, the chromosome movement during meiosis had been worked out. That is by Walter Sutton and Theodore Boveri. That is chromosomal behavior. Behavior of the chromosome. Behavior of the Walter Sutton and Theodore Boveri. Behavior of the chromosome was parallel to the behavior of genes. And it is used in the chromosome movement. These are things that we explain. Next class we have to explain all these things here in the case of to explain the Mendel's laws compared to this chromosome movement to explain the Mendel's laws. And you have studied the behavior of the chromosome during mitosis, equational division, and also during meiosis, reduction division. The important things to remember that chromosome as well as genes occur in pairs, and the two alleles of a gene pair are located on the homologous sites on homologous chromosome. Homologous sites on homologous chromosome. Okay, but then now we have to stop here. Okay, but next class onwards we have to continue this chromosomal basis of inheritance. Chromosomal basis. Okay, then we go to other sites, that is in the case of these disorders and all other things, pedigree analysis, and all human disorders, all these things, mutation, etc. That will be studied later. Or sex determination, all these things will be taken in the next class. Yes, now you stop here. Okay, thank you everybody.